So these are the chemical elements present in the protein. But why our body need a protein? And why are we making a protein? Yes, its basic building block is the amino acids. So basic building block of the protein is amino acid. But why our body needs a protein? Again, the same question. देखा यही पढ़ा था हमने बस यही पढ़ा था that the protein, it is needed for muscle growth, for the tissue repair. So the protein is also present in the DNA, messenger RNA. That was the formation of the protein synthesis. Ji beta, the one you are telling me, it is the protein synthesis. Do we need a protein anywhere elsewhere in our body? Do we have a protein? Antibodies, very good. So we have antibodies as well. We have hormones. We have enzymes. You have your hair made up of protein, which is keratin. The keratin in your hair, it is also made up of protein. Then you have protein present in RBCs as well, that is hemoglobin. Then you have a protein in your skin, that is collagen, that is used to strengthen the tissue. Then you have proteins in the muscles, that are actin and myosin. These are the proteins which are present in the muscle. And anything else which I may have skipped? You have your cell membrane, it also contains protein. Then you have the storage protein as well. And what are the storage protein? Like we have casein. We have the casein protein, which is present in the milk. We have ovalbumin, which is present in the egg white. That is why we say that egg is a very good source of protein. Why do we say? Because it has a storage protein, ovalbumin. So that is present in the egg white. These are the places where our body, it needs a protein, and the proteins are needed in all these parts of the system. Now you said that proteins, they are made up of amino acids, right? So amino acids, these are the basic, we can say that amino acids, it is the building block of protein. But do you know that in our body, ovalbumin is the protein which is present in the egg white, the egg that we consume. So the whitish part of that egg contains a protein, ovalbumin, that is needed by our body. Okay, as we say that amino acid, it is the building block of the protein. So do you know that our body, it contains only 20 different types of amino acid? So only 20 different types of amino acids are present in our body. They are not much amino acid present in the body. There are only 20 different types of amino acid that are required. When they are joined together, they form different proteins which are needed by our body. So you will see only 20 different types of amino acid which will be joining together, making different kinds of protein that is needed by our body. So what is the basic structure of the amino acid then? So the basic structure of amino acid is that amino acid, it contains a carboxylic group on one side and amino group on the other side. And with that, it also contains the third thing, which is the R group. And what is the R group? R group is the side chain. That side chain, it could be any group attached with the amino acid, and it will form another type of amino acid. So the first thing that we need to understand, that in every amino acid, in every amino acid, you are going to see one carboxylic acid group. It is mandatory for every amino acid to have carboxylic acid group. Then it is mandatory for every amino acid to have amino group. Now it does not make any difference if you are going to shuffle these groups, but these groups should remain opposite to each other. Like you cannot change the direction like this. You have to make sure that if the carboxylic acid is on the right side, then amino group should be on the left side. If carboxylic acid is on the left side, so then amino group should be on the right side of the amino acid. Then the placement of the R group. The R group should be present perpendicular to the carboxylic acid. What is perpendicular? Perpendicular means 90-degree angle. So it means that the carboxylic group is over here. So R group should be perpendicular to the carboxylic group. So let's see again. Let's see it again. So this is, we have carboxylic group. We have one carboxylic group. We have an amino group. Then we have the third one as the side chain, which should be perpendicular to the amino acid. Okay, now the side chain, it could vary. It could have different number of carbon atoms attached to it. So in the exam, if you're being asked about the basic structure of amino acid, so the three things should pop up in your head. The first thing that it has carboxylic group, and the second thing it has amino group. They both should be on each side. Okay, they should not be up and down. They should be on the sides of the amino acid. Then we have R group, which should be perpendicular to the carboxylic group. Now the next, let's see how many different types of R group can change the amino acids. So we have the first amino acid. Is it clear? The structure, the basic structure of amino acid. Is it clear to all of you? Okay, so let's see the examples of the amino acid, and you will see the difference in the R group now. So everything is going to remain same. If I'm going to take the three examples of amino acids over here, I'm going to take the same structure of amino acid. The same structure is that it is having the carboxylic group over here, if you see here, and it has amino group over here. Okay, the only thing that I'll change will be the R group. So let's exact copy paste these three structures over here. Iman, is it clear to you? Okay, so I have copy pasted exact same structure. Now I'm just going to change the R group. If you see the R group over there, so I'm just going to change the R group. So first of all, if I'm going to replace the R with this group, so it will be called as, the amino acid is called as alanine. So we will form an amino acid which is alanine. If I'm going to simply replace the R group with the hydrogen, so we can say this amino acid as glycine. If I'm going to replace the R group with the carboxylic group, again C, H, and double O. Okay, so I can call this amino acid as serine. So this is how changing the R group can change the type of amino acid. Clear? So what have we changed? Only the R group. So carboxylic group and the amino group, they are going to remain same in every amino acid. The only thing that is going to be changed is the R group. Now let's see the glycine before I move forward. नहीं नहीं आप लोगों ने लर्न नहीं करनी, आपको आएंगी पेपर में बनी भी आएंगी. ओके, आपने उसके अंदर डाइपेप्टाइड कंडेंसेशन रिएक्शन करना होगा, मैं भी सिखाती हूँ वो कैसे करना है. ओके, अच्छा, तो लेकिन आपको जैसे स्मॉलेस्ट है ना, जैसे मैं बताने लगी, ग्लाइसिन इज द स्मॉलेस्ट अमीनो एसिड among 20 amino acid. Glycine is the smallest one, is the smallest amino acid. Done. Let's start. Let's see how can we form a dipeptide bond. How are we going to form a dipeptide bond? So we can form dipeptide bond. If you remember the enzymes chapter that we did in the O levels, or if you have the basic knowledge, Iman, so we know that when we join two amino acids together, they are joined together through a dipeptide bond. Or you must have studied about the peptide bond. Forget about the di word. Okay, you must have studied about the peptide bond. That peptide bond is the bond which is used to join two amino acids together. So when two amino acids they join up together, they form a peptide bond. So we need to look into it. We need to see that how they are going to join two amino acids together. How the two amino acids, how the bond of the peptide is formed between two amino acid molecules. So again, we are going to study about the condensation reaction. And what was the condensation reaction? It was the removal of water molecule. So, but we need to know that from where, which position we are going to remove the water molecule. Condensation reaction हमने lipids में भी किया. Condensation reaction हमने carbohydrates में glycosidic linkage किया और lipids में हमने ester bond formation की. Now today we are studying the peptide bond formation through the same condensation reaction. But the nature of the bond, but the bond which is formed between two atoms, it will be different. So the condensation reaction is the removal of water molecule. Now we need to see that from where we can remove the water molecule. So let's take two different amino acids together. So let's take glycine and alanine. So I'm going to take the alanine and the glycine over here, and we will join these two amino acid molecules to form a peptide bond. This is alanine, this is glycine, and I'm going to join the glycine with the alanine. This is glycine. Now I'm going to join these two amino acids together. As we know, we have studied earlier that condensation reaction is the removal of water molecule. So now we have to remove the water molecule. So we have to see that from where we can remove the water molecule. We can remove the water molecule when we will remove OH and H. So we are going to remove OH and from the carboxylic group and the H from the amino group. So from where I'm removing OH from the carboxylic group. So we are going to remove OH from the carboxylic group and we are going to remove H from the amino group. So when we are going to remove these OH and H from each of the sides, minus water. So let's join them together. So I have removed OH and I have removed the hydrogen from the amino group. I'm going to bring it a little closer. And from here we are going to remove. So can you see that we have formed a peptide bond here? this is how we can form a peptide bond. And this is called as dipeptide. And the structure that we have formed over here, it is called as dipeptide. This structure is called as dipeptide because it is joining two amino acid molecules together. So we can conclude from this that a peptide bond is formed only when we are going to join, when we join OH group from carboxylic acid is removed with the hydrogen group from amino acid. So only then we can form a peptide bond. Okay, let's make another peptide bond. Now you have to make a peptide bond between serine and glycine. Serine and glycine. So you have to make a peptide bond between a serine and glycine. NH, H. H and H is the glycine. So when we are going to join the glycine with the serine, which is N, H, H, C and C, double bond O and OH, H and C, double bond O, H and H over here. Yes. So when we make a peptide bond over here, this is very important for you to understand that where we can make a peptide bond. We cannot make a peptide bond between the hydrogen of this and the oxygen of this. No, we cannot do that. We can never make a peptide bond with the R group. Okay? If the R group is containing carboxylic acid as well, we cannot make a peptide bond with the R group. This is what you have to understand. Because exam में आपके पास different structures आते हैं, they want you to be confused. So you just have to remember that a peptide bond is always and always formed between amino acid and the carboxylic acid group. Amino acid of one, amino group of one amino acid and the carboxylic group of other amino acid. It cannot be formed with the R group. Okay? ये आपको कंफ्यूज करता है एग्जामिनर आपको स्ट्रक्चर देकर पूछेगा इज इट अ पेप्टाइड बॉन्ड, इट इज नॉट अ पेप्टाइड बॉन्ड, क्योंकि पेप्टाइड बॉन्ड कभी भी R के साथ साइड चेन के साथ फॉर्म नहीं हो सकता. इट इज ऑलवेज हैव टू बी फॉर्म विद द carboxylic acid of one amino acid and the amino group of the other amino acid molecules. Okay, so we can never form a peptide bond with the R group. चलें, we can form this peptide bond again over here through the condensation reaction. मुझे लगता है परीक्षा हमें बनाकर भेजा है. जी बेटा, परीक्षा मैं एक सेकंड यहाँ पे अटैच कर देती हूँ so that all of you can see. Yes. Can you guys check this out? Is this right? Is this a right peptide bond being formed? Is this a right peptide bond being formed? H is missing. In glycine. Okay. चलें, let's correct it. यहाँ नीचे भी H missing है, मुझे तो ये वाला H missing लग रहा है. Yes, here this H is missing and here this H is also missing. ठीक है परीक्षे? कार्बन की वैलेंसी हमने 4 कंप्लीट करनी है, ठीक है? So these H were missing. ठीक है. So what we concluded from this discussion that R group is never involved in formation of peptide bond. So we have never included the R group in the formation of peptide bond. Clear? So you know how to make a peptide bond now. You know how to make an ester bond now. And you also know that how to make a glycosidic linkage as well. So you know all these three bondings. How can we make an ester bond? How can we make a peptide bond? And how can we make a glycosidic linkage? And from today, you will know that peptide bond is always formed between OH of carboxyl and the H of N amino group. Okay, so we can say the dipeptide is a peptide that has two linked amino acids. And what is a polypeptide then? What is a polypeptide then? We can call it as a dipeptide. And it is containing a sequence of amino acid. It has two linked amino acids. Yes, a sequence of amino acid is called as polypeptide. So polypeptide means simply that a chain of amino acid that is linked together. For example, I'm going to link a glycine. I'm going to link alanine. I'm going to link serine. I'm going to link the glutamic acid as well, aspartic acid, arginine. So this will make up polypeptide. So a chain of amino acid that is linked together by peptide bond. Okay, I hope that by the end of this, you know how to make a peptide bond. All of you. Iman, Fatima, Aisha, Barishe, all of you are well aware how to make a peptide bond now. Okay, so we are starting another topic now. And that topic is very important for you to understand. And it is about the types of amino acid. So how can we, types of the protein in fact, how can we classify the protein into different types as per their structure? We know that protein, when the polypeptide is going to join together, so it is going to make a protein structure. But it is not that much simple that we think. There are different structures of the protein. So in the total, we are going to study 4 structures. Is it peptide bond or dipeptide bond? No, it is peptide bond. The one bond is peptide bond. But when the two amino acids they are held together by a peptide bond, it is called as dipeptide. ठीक है, जैसे हमने polypeptide किया, so it is dipeptide. This one is dipeptide. But the bond here is a peptide bond. Okay, Aisha? The bond is peptide bond. But when two amino acids are joined together through peptide bond, we call these amino acids as dipeptide, like we did for the disaccharide, and then we did polysaccharides. Okay, so we're discussing about the protein structure, and in the protein structure, we are going to see four different types of the structures of the protein. And what are those four different types? The one is primary structure, the other one you will see is the secondary structure, then tertiary structure, and then you are going to see about the quaternary structure. अब जैसे जैसे आप इनके नाम आपको थोड़े से complex लग रहे हैं, so they are a little complex, but if you will keep a pace with me, so इंशाल्लाह आपको difficult नहीं लगेंगे. So starting from the primary structure, the primary structure is very easy structure that you all have studied right now. Primary structure. Okay, so what is a primary structure? Primary structure is just this structure. This structure is called as a primary structure. बस यही structure है miss? Yes, बिल्कुल यही structure है. This is the only primary structure that in which we have sequence of amino acid in a polypeptide chain. Primary structure is a structure that refers to the sequence of amino acid in a polypeptide chain. ठीक है, this is the primary structure, that is it. अब इसका sequence जो है, that you know that the sequence of amino acid is going to give you the type of the protein and the structure and the function of the protein. Like if we have different amino acid, if I have this glycine, then again aspartic acid, if I have alanine, if I have serine, if I have again glycine, if I have serine over here. So the sequence of amino acid that is present in this protein, it is going to determine the type of the protein. So why are we emphasizing on the sequence of amino acid? If you remember when we did the protein synthesis, so we emphasized it over there as well, that this sequence is sent by the DNA through messenger RNA. Okay, so this sequence of amino acid determine the structure and type of protein. That is why we have different enzymes. That is why the protease differs from the lipase, and lipase differs from the carbohydrates. So that is why we have different sets of enzymes, because they have different sequence of amino acid. And if you remember, this was being asked in the past paper question, that why the sequence of amino acid determines the type of the enzyme, because the sequence of amino acid is going to tell you that which protein is going to be formed and what will be the structure of that protein, and what as per its structure what function that protein is going to do. Okay, type, then structure, and then what is the function that it is going to perform. So it determines the structure, type, and the function of protein. Okay, is this clear? So if we are going to change only one, if I'm going to change only this one protein, and I'm going to put any alanine over here. If I'm going to change only this sequence, one amino acid है ना, ये मामा सिर्फ एक चीज खानेंगे तो क्या फर्क पड़ जाएगा? अगर हम सिर्फ एक amino acid को change कर देंगे तो क्या फर्क पड़ जाएगा? It is going to change the whole protein. Even one amino acid is going to be changed, so then it is going to change the whole protein and its chemistry. So we need to be very particular about the sequence of amino acids. चलिए, ये था primary structure. Let's move to the secondary structure. Now, in the secondary structure, we also have a long chain of polypeptide or a long chain of amino acid, but it is too much long. So we need to compact it. When it is too much long, so the chain by itself, it starts coiling. And when it starts coiling, it starts forming bonds as well. So let's see that which bonds it's going to form. So in the secondary structure, we refer this structure as the folding or coiling of a polypeptide chain. Polypeptide chain due to interaction between amino acids. Okay, so we are going to see the folding or coiling. अभी टेंशन नहीं लेनी, अभी मैं सारी folding और coiling आपको बताती हूँ कैसे होती है folding और coiling. So for example, we have a very long chain of the polypeptide, और इतनी long chain के अंदर हमारे पास it started folding. ठीक है, so let's see how it is going to start folding. First of all, that we have a long chain of amino acid and it starts folding. This is called as folding and the coiling of amino acid, but it is not that simple. So let's dig into its chemical structure, and we will see that why it is being folded and why, if it is being curved or it is folded, so what is the reason behind it. So let's see the reason behind it. For example, if we have this one amino acid molecule over here, and in this amino acid we have this nitrogen, we have this hydrogen, and with this amino acid we have, okay, so we have this peptide bond, then we have amino acid 2 over here, and with this amino acid we also have carboxylic group, we have amino group over here, and again we have the third amino acid, and with the third amino acid we also have peptide bond over here. So again we have NH, and this goes on, okay. So this is going to be turned around, it will be keep on going. Okay, what happens over here is that this hydrogen, it is having a partial positive charge, and this oxygen, it is having a partial negative charge. So if I just coil this section like this, okay. So this hydrogen is having partial positive charge and this oxygen is having partial negative charge. So what is going to happen, that hydrogen bond is going to be formed between two molecules, between the hydrogen of amino group and the oxygen of carboxylic group. Already we had the peptide bond, ठीक है? हमारा peptide bond बेटा कहीं नहीं जा रहा. We have the peptide bond over here. we are not going to forget about the peptide bond. Obviously, the amino acids, they are joined together through the peptide bond. But there is another bonding that amino acids start doing when it is enlarged enough, when it has a number of amino acids in one chain. So when it starts coiling, so it starts interacting with the positive and the negative charges. So the hydrogen being having positive charge and the oxygen being having partial negative charge, so it will form a hydrogen bond. So hydrogen bonds are formed between amino group of hydrogen and oxygen group of carboxylic acid of amino acids. So let's see in the chain, how are they going to form the hydrogen bond? For example, this is what they are going to form. Then again, if we move down, so this is how they are going to form hydrogen bond. This is called as secondary structure of amino acid, but it does not stop over here. Why it is not stopping over here? So if you see this primary structure, we can refer these bonds as, so if you see in this structure that I have made, that in this structure you are going to have the spiral and the foldings and the spiral. Then again the spiral and then the foldings. So this structure is having foldings and the spiral that we have seen in this definition. So you have studied in this definition that the secondary structure is a structure that has a folding or coiling. So here is this coiling and the folding. Can you see this coiling over here and this folding? But the problem is that you need to know that what is the chemistry behind this coiling and folding, and what do I need to understand from this coiling and folding. So what are we going to do? That we call this coiling structure as the alpha helix. Now what is alpha helix? What is alpha helix? That when the hydrogen bonds, they are formed in this manner, so we call it as alpha helix. Okay, so we call it this structure as alpha helix, and the alpha helix structure is going to make a bonding, is going to make a coiling. Then we call this beta pleated sheets. What is the beta pleated sheets over here? What is the beta pleated sheets? That it is going to form the hydrogen bonds in just like a pleated shape. Okay, this is called as beta pleated sheets. So we are always going to show the alpha helix in the exam. You are always going to show alpha helix. This is a representation that you need to know, that this is how you are going to show the alpha helix. And how are you going to show the beta pleated sheet? So you are going to show the beta pleated sheets like this. Okay, like this. Now let's do it. Let's make a secondary structure now. So I'm going to make a secondary structure using the foldings and the coiling. Okay, I'm going to use foldings and coiling. First, we have seen that what is folding, what is coiling. Then we have undergone a structure that it has some different arrangement of the bondings, that is alpha helix and the beta pleated sheets. Then we know that how are we going to represent the alpha helix and the beta pleated sheets. So we are going to represent the alpha helix and the beta pleated sheets in this manner. So let's make this alpha helix and the beta pleated sheet like this. So I'm showing this beta pleated sheets in the arrow. And then I'm going to move forward and I'm going to show you the alpha helix in the coiling manner. Then I'm going to further move forward and I'm again going to make the alpha helix over here. And then I want to show the beta pleated sheet over here. So I will show the beta pleated sheets like this. Like this. And we have these two ends free and like this. Okay, so these ends are, they are ready to pair with another polypeptide chain. Okay, the one that I have shown you in the dots, that they are again ready to form a bonding with another structure, with another polypeptide chain. I hope it is clear. So you need to know that these hydrogen bonds, that we studied, these hydrogen bonds, they are strong, but not that much strong. They are strong, but they can be easily broken down by high temperature and high pH. So आपके पास, आपके mind में फौरन से आना चाहिए था, oh, this is talking about enzymes. That is why the enzyme gets denatured at higher temperature and the higher pH. That is why the active site shape is deformed. ठीक है, अब मैं बताती हूँ कैसे active site shape is deformed. It is by high temperature. वैसे तो हम enzymes में पढ़ेंगे, but I'll give you a little sneak peek of it. High temperature and high pH. आज तक आप लोग पढ़ते आए कि enzymes की active site है, ठीक है? This is the active site of the enzyme जो आपको ये नजर आ रही है, ठीक है? इन active site deform कैसे होती है? बेसिकली ये जो एक्टिव साइट होती है ना एंजाइम की, प्रोटीन से बनी हुई है, सो पार्शियल पॉजिटिव पार्शियल नेगेटिव चार्ज होता है, सो ये क्या करती है, आपस में ना हाइड्रोजन बॉन्डिंग बना देती है ऐसे। जैसे ही हाई टेम्परेचर होता है, ये हाइड्रोजन बॉन्ड ब्रेक डाउन करता है और एंजाइम जो है आपका वापस अपनी एक्टिव साइट को डीफॉर्म कर लेता है, ठीक है? सो हमेशा एक्टिव साइट जो डीफॉर्म होती है, दैट इज डीफॉर्म बिकॉज ऑफ द ब्रेकेज ऑफ हाइड्रोजन बॉन्ड्स व्हिच आर प्रेजेंट इन द एक्टिव साइट ऑफ द एंजाइम। सो जब ये हाइड्रोजन बॉन्ड्स ब्रेक करते हैं हाई टेम्परेचर एंड पीएच, सो द एक्टिव साइट इज डीफॉर्म, इट लूजेस इट्स शेप। सो समझ नहीं आता व्हाट डू आई डू, सो वो जैसे हाइड्रोजन बॉन्ड बनते हैं, सो इट मेक्स द शेप अकॉर्डिंगली। Okay, so that is why the enzyme gets denatured at high temperature and high pH. Clear? हाँ, अभी पढ़ेंगे। एंजाइम्स का चैप्टर बहुत इंटरेस्टिंग है। बहुत मजा आएगा आपको एंजाइम का चैप्टर पढ़ते हुए। बस वी आर गोइंग टू एंड दिस चैप्टर और फिर हम इंशाल्लाह विद इन 2 डेज एंजाइम स्टार्ट करेंगे। So is it clear to all of you? I'll send you these notes so that you can revise these two structures of the primary and the secondary structure. क्योंकि tertiary और quaternary structures में कुछ bondings increase हो जाएगी. So if you have recalled the previous knowledge enough, then you will be able to catch up the next structures. Okay बेटा?